This is Dr. Lauren Launen, continuing with the lecture set on DNA replication. We just finished discussing the subunits that are found in DNA polymerase III in E. coli. You should know how many subunits they are and have a general understanding of how many genes would be required to produce that enzyme and what the different bits and pieces are. So let's go back to this term holoenzyme, which I mentioned a moment ago. A holoenzyme means the entire assembled functional enzyme. DNA polymerase III and also RNA polymerase are examples of holoenzymes. This means that you have to have multiple polypeptides together, working together to get a functional enzyme. It's a term that is relevant to enzymes alone. Okay? So for both DNA and RNA polymerases, this includes a requirement for something that is not a protein. It's a small cation, and that is the cation magnesium. It's a 2+ charge. So magnesium in this case is a coenzyme, and magnesium is shown. This is a zoom in on the beta subunit within the entire DNA or RNA polymerase structure. And these little green blobs are the magnesiums. And if you don't have them in there, the protein won't take the correct shape and therefore won't be able to do its job. In this case, the beta subunit wouldn't be able to help DNA polymerase III clamp on to the DNA strand, and so synthesis would not occur. And there's a practical reason I'm pointing this out, because if you're actually doing PCR reactions, if you were to set a PCR reaction up, which is DNA synthesis in a test tube, and you forgot to add the magnesium, then your reaction would not work. So it's important. All right, let's talk for a moment about the initiation of DNA replication, the beginning. How do you get started? So initiation of DNA synthesis or replication begins at sequences that are called the origins of replication. Synthesis will begin at those locations, and then it will move bidirectionally, meaning in either direction. Okay? The origin sequences are sites on each chromosome, and they are specific nucleotide sequences whose job it is to attract the replication proteins or enzymes. Origins are very well conserved, so if you look at the origin of replication within an E. coli, it looks almost identical to the origin of replication in a Streptomyces bacterium or a Streptococcus bacterium or any other type of bacterium. And similarly, if you look at the origins of replication that are found on our human chromosomes, they'll look a lot like the origins of replication that are on chimpanzee or dog or cat chromosomes. Here's a picture of the origin of replication site on E. coli, and it is actually named the ORI-C site. Here's the E. coli chromosome. Like most bacteria, E. coli has one circular chromosome in its genome. And the ORI-C site, which isn't really drawn to scale here, is in total 245 base pairs long, and it looks like this. It has repeating structures, red repeats and blue repeats. They're not literally colored in the chromosome, they're just colored on this figure. So the red repeats occur as 3 sets of 13 nucleotide long sequences, and the sequence looks like that. We know what it looks like. It's been characterized. Similarly, these blue repeats happen 4 times, and they look like this. So we call these tandem, meaning side-by-side, repeats that are arrayed or placed together, and they are located like this in the origin of replication site. So if you look at any E. coli chromosome, if you zoom in on the chromosome and you look at all the sequence data, you'll be able to search for an area that has these patterns, and you'll know that when E. coli is getting ready to go through cell division, it's going to need to copy that chromosome, and the copying is going to begin at that site, the ORI-C. Here's what it would look like, or a cartoon schematic of what it would look like. This is an electron micrograph showing what it actually looks like. Okay? But if we look at the cartoon, there's the ORI-C site, and then on the opposite side of the chromosome, there's a site called the TER site, and that's the site where where synthesis will actually stop. Okay? So bacteria will always have one origin on each of their chromosomes. Most of them only have one chromosome. And then the DNA replication proteins will land there and they will start synthesis, and they'll go in either direction. One set will go this way, one set will go this way. And that will create this structure. And then a few moments later you'll see this. And then ultimately the sets of proteins will bump into each other at the terminus site, and then you'll have the resolution of two new chromosomes. Each of those would be within a cell, right? And then the rest of the cell would finish forming, and you would have cell division, which is called cytokinesis. Each of these spots will be a replication fork. One replication fork here, one replication fork there. All right, so situation's a little bit different, but mostly similar in eukaryotes. Eukaryotic chromosomes are, of course, linear, and they occur in pairs, but that's not super relevant right here. The linear is, though, they're also very large compared to bacterial chromosomes for the most part. So that means that you have to have many origins of replication, otherwise DNA synthesis would take too long to be practical for the cell. So here is one eukaryotic chromosome, and within it there will be multiple, like here in this figure, 1, 2, 3 origins of replication. Those sites are going to attract the replicating enzymes and the DNA polymerase, for example, and then synthesis will start in either direction, this way and this way. It's bidirectional. Here's a replication fork there, here's a replication fork here. Okay, similarly there'll be a replication fork here and here. And there'll be a replication fork here and here. Synthesis will go this way, and synthesis will go this way until they bump up into each other, and you've got two chromosomes formed from what used to be one. Next, we're going to discuss the idea of the replication forks. So we're going to zoom in on the replication fork. So here's the replication fork, and remember, in the actual replication structure, which are often called replication bubbles, that would be the replication fork there or here. So replication forks travel, and there are many of them. Okay. This figure is showing you, a replication fork in one particular orientation, but over on the left side of the screen, not drawn, there'd have to be another one. Okay. So the pink is the old template molecule, and the blue is the new molecule that is being synthesized, and it's going to always be synthesized complementary and anti-parallel to the template. So we've created a replication fork, right? And because of the orientation of DNA, you're going to have to make the new DNA in the complementary but also anti-parallel orientation. So that means that since DNA synthesis always happens 5' to 3', right? Adding nucleotides onto the 3' end. The 3' ends of the new strands are marked with the arrows in blue here. You're going to have new DNA synthesis that's traveling both into the replication fork and away from the replication fork at the same time, mediated by the same enzyme. Okay? So we use some naming here so that we can study and understand these processes better. We talk about the new strand being synthesized moving away from the replication fork. We call that the lagging strand. And any strands that are being synthesized going into the replication fork are referred to as the leading strands. So leading strands are going to go into the replication fork. Lagging strands are going to be going away from the replication fork. You should be able to figure out, if I give you a picture like this and I don't label the ends, right? Say I don't label this 3 or this 5, you should be able to figure out from the template strands how to label the ends of the new strands, how to label the direction of synthesis, right? And how to name them lagging or leading. That's something you should be able to figure out. What we're going to get to in a moment, we're going to talk about leading strand synthesis, and then we're going to talk about lagging strand synthesis, which happens in bursts. It's called discontinuous synthesis. And we're going to break those apart as if they're separate processes, but they're actually happening at the same time in nature, mediated by one DNA polymerase enzyme doing the work on both strands.